morning. Box. Box is a recording device, so good morning, Box again. Since this is environmental biology and our environment has changed quite a bit, I want to talk to you about viruses, the coronavirus especially, okay? And we'll get a paper done on the coronavirus, how it has impacted you, what are you doing to limit the spread, is there anything that can be done to limit the spread, etc. That'll be your paper. But some facts about viruses, okay? The two types of viruses. There's a DNA virus and an RNA virus. Coronavirus is an RNA virus right here, okay? But let me talk to you about the structure of a virus, okay? This will be your first objective, and I'll put this list on this sheet on the web or email it to you. I'll do that probably this morning or in the morning, okay? This will be due next week, and then we'll have one more topic after that, and that'll be the course of this semester. Okay, a virus, composition. That's all there is to a virus. There's some genetic material on the inside. That's what this little squiggly line right here represents, either DNA or RNA. And the covering right here is a protein coat. So it doesn't really matter about how many DNA bases. I saw 27 kilobases or something like that. I don't know. I looked up something about it. I don't really know. I know it's a fairly large RNA virus, but this is an RNA virus. It's not a DNA virus. So here is a virus. Now, how do they replicate? They have no mitochondria. They cannot produce any energy at all. So they have to fasten to a host cell to replicate. Without a host, they're dead. Now, or dormant. They can be dormant for quite some time. I don't know the length of time on that. Heard the other day they could live 24 hours on cardboard. They got to be exposed. The cardboard's got to be exposed to it. So somebody infected has to breathe on the cardboard and it stays on it for 24 hours. Somebody else touches it, and then all they do is touch their mouth, their lips, their eyes, and it gains a portal into the recipient. So here is a virus right here, and I'm going to take a look at how it replicates. All right, this will be our first objective here. So this is a host cell. This is a cell membrane. This is a respiratory tract. This is a cell in the respiratory system right here, okay? This is a host cell right here. On the cell membrane, there's a receptor site for the virus. This is a virus. This is a receptor site on the host cell membrane. All right? Now, without the receptor site, the virus cannot attach. It cannot enter in. Now, when the virus attaches, it's the genetic material that enters into the cell. It's not the protein coat, the capsid. It's not that at all. It's the genetic material inside the virus. So if it were DNA, it would simply match up to our host cell DNA and our chromosomes in our nucleus right there. Whenever the cell replicated, the virus would replicate. But this is an RNA virus right here, okay? So the RNA virus invades the host cell, right? So here's the RNA virus. Let me go ahead and make this up to give this a bigger color right here. Excuse me just one second. I thought to pick a piece of red in here. So it's an RNA virus. That's genetic material. I don't know if it's a single, well, RNA single strand, DNA is double strand. So here's a strand of RNA. Remember, it's got uracil. I don't need to know the difference between DNA and RNA. Here's an RNA virus right here. And inside the host cell is the RNA. Now, RNA cannot replicate itself. It simply cannot do it. It has to become DNA. So this RNA is changed to DNA in the host cell by an enzyme called RNA transcriptase. Without this enzyme in our host cell, the virus is dead. So this is RNA. Has to become DNA. Through an enzyme called reverse transcriptase. I want you to know this. This will be part of your answer. Reverse transcriptase. REVERSE TRANSCRIPTASE. Reverse transcriptase is this enzyme that converts the RNA to the DNA. Now the DNA can attach to our DNA in our chromosomes, and it does it. Excuse me. Gets inside the nucleus, attaches to the host cell chromosomes, And whenever the host cell replicates, the virus replicates. That's the way the virus survives, okay? Now, that's the objective of the first component. There's the components of the virus, genetic component, some kind of DNA or RNA along a protein coat, a capsid. How do they reproduce? The RNA has to be converted to DNA through the reverse transcriptase, all right? Now, how might this particular coronavirus have been transmitted to a human? Now, since this is a very large coronavirus, now coronaviruses are around anyway. They've been around for, I don't know, hundreds of years. They cause a common cold. But there are some that are more virulent than others. This is one of them. This may be the most virulent coronavirus, maybe the most virulent, I don't know about the most virulent virus, because Ebola was very virulent. Okay, this is a very virulent virus. So how might it be transmitted to a human? Well, I started to say I don't really know, and I don't know, but let me give you a guess. It's present in bats. And in China there are a lot of bats. Hell, in the US there's a lot of bats right there. So it's thought to have been transmitted to a human through a bat bite. Now this is kind of scary, since if it's in one bat, it's in thousands of bats or millions of bats right there. So there's a reservoir for coronavirus in bats and other mammals that also, cows get it right there, chickens get it right there. So evidently, maybe, and I need to emphasize the word maybe right here, maybe through the bite of a bat, of an infected bat to a human, and it mutated somehow, so it's transmitted from human to human. This happened to occur in China. I don't think that the Chinese made the virus. I don't think that they did, okay? Although you can make a virus, but I don't think the Chinese made this one. Okay? What's the time between exposure and symptoms? Well, this is the scary part. Normally we think about, 7 to 10 days of exposure, where there's a quarantine of 2 weeks, and maybe it is 2 weeks, maybe it is up to 2 weeks right there. But I have heard of being healthy one day, 8 o'clock one morning, being on a respirator at 8 o'clock that night. So as short as 12 hours, that's what I want you to put down. As long as maybe 14 days, as short as 12 hours. That's a hell of a variation right there. And I don't have a good explanation for you, okay? But I do know that we can pass viruses on from cell to cell without having the symptoms of the disease itself, because we are avirulent. Avirulent means without any symptoms right there. But when we become virulent, that means the virus is rapidly dividing. The host cell makes a lot of viral particles, not one cell to one particle, one cell to thousands of viral particles. Then the cell dies and the virus is released, and then many other host cells are infected. And this sucker's pretty bad because it causes a lot of damage to the lungs, as a respiratory virus would. Okay? Now, what are the symptoms of this virus? It can be very mild. If you're avirulent, you have no symptoms whatsoever. No symptoms whatsoever. I don't think I'm a carrier. I hope I'm not. I don't know. I haven't been tested. But I would have to have some kind of symptoms, a fever, a cough that develops suddenly within a few hours, okay? I don't know about diarrhea, maybe in some people right there, but certainly a fever, respiratory problems, labored breathing, a fever. Those would be two major symptoms of, and a cough, let's make them three major symptoms for you to expect, suspect a viral infection. But it's going to be a rapid onset within, once the fever starts, once the cough starts, within 8 hours, 10 hours, somewhere in there. Okay? So keep that in mind. Okay. What were the treatments of the infection? Well, if this were a bacteria, it'd be antibiotics. It would be that simple. Antibiotics would wipe the darn thing out. But antiviral drugs are really, really tough to get a hold of. And I don't know how good they are anyway. Okay, they're not very, there hasn't been a whole lot of experimentation on antiviral drugs, okay? Not to the extent as far as antibiotics. Antibiotics have been around for 100 years. But antiviral drugs have been around maybe 20 years, and they're just not that effective, not yet. So we have to treat the symptoms, have to treat the symptomatic, okay? But the main way that you can treat this thing is you got to breathe. Maybe it's a respirator, maybe that's what that it is. You got to be taken care of. You got to be, somebody's got to look after you. Got to, because you probably can't get up, so you're going to be hospitalized. So you look up the treatments for the virus and help me out on this one right here, okay? Now, my last objective on this, and this is a short series right here. How can serum from a person that has recovered from an infection be used in treatment of an infected person? Affected and infected, okay? The serum contains antibodies. Let me tell you about an antibody right here. Whenever we are exposed to something foreign, we call that foreign substance an antigen. We make antibodies against it, okay? Antibodies are proteins. We make antibodies against it. And the antibodies will fasten to the antigen, and it may neutralize it by itself. It may precipitate, it may speed up phagocytosis, being engulfed by our own immune cells right there. It may inactivate the virus, okay, in this case the virus itself right here. So an infected person that recovers, you got to recover, because if you're dead, you ain't going to make the damn antibodies. An infected person, or a recovered infected person, is going to have antibodies in their serum. So you can donate serum. I never have when I go to the Red Cross. I just donate whole blood, but I could donate my serum. Years ago when I was in vet school, I got vaccinated for rabies. Paid $150 for 3 shots. Not $150 a piece, $50 a piece for 3 shots. And I would go to Atlanta. I was in school in Athens, in Georgia. I would drive to Atlanta and sell my serum. On the way back, I'd stop, pick up some beer, and have a good time. I remember selling my serum because I had anti-rabies antibodies. And rabies is a virus also. So I am sure that, and I don't know why this wasn't thought of earlier. It's probably been done for 50 years, maybe 100 years.